In this tutorial we'll look at another genetic tool referred to as the use of restriction enzymes or restriction endonucleases. These are special kinds of enzymes that have been found in various bacteria and have been used now for many years in assisting with genetic engineering, and we'll look at how they're used in another tutorial. But let's just look at what a restriction enzyme is and what it does. So it is a genetic tool. So that is, it's a tool that geneticists can use to manipulate other DNA. So what is a restriction enzyme? A restriction enzyme is an enzyme that can cut through DNA at a specified base sequence. Alright, so it's an enzyme that can cut through DNA, at a specific base sequence. Cut in abbreviated commas because it doesn't really cut, it breaks the bonds between various bases, hence separating the DNA molecule. There are two essential ways that restriction enzymes work. One is to cut straight through between two pairs of bases, as shown here, and this process creates what are called blunt ends. We'll have a look at that in a moment. And the other way that a restriction enzyme can cut DNA is in a staggered cut, and this creates what's called sticky ends. And these are the ones that are most useful as genetic tools. So a restriction enzyme, here's an example here of ALU1, and we have the 5' end of the DNA through to the 3' end, and then the other strand 5' through to 3'. So this is the double-stranded DNA molecule. These are, there'll be millions of base pairs, but what's important to note here about restriction enzymes is they will only cut through at a specific base sequence, that is a recognized base sequence. So ALU1 only ever cuts between G and C, or CG on the other side, in a sequence AGCT. So it will only ever cut straight through at that point. And it creates a blunt end. The blunt end refers to the fact that you have your DNA has an even ending if it cuts straight through the middle. So you have a GC and you have a CG, and then these are separated in the middle, so it's a blunt end. This is contrasted with something like Echo R1. Let's have a look at this one down the bottom. Echo R1 is a famous genetic enzyme, restriction enzyme. It comes from E. coli, strain R1, and it creates what's called a staggered cut and therefore sticky ends. Echo R1 will only ever cut where the sequence GAATTC is seen, and it cuts between the G and the A at one end and the A and the G at the other end. So it's a staggered cut. So this results in DNA that looks like this. So one piece of DNA will have a long single tail like that, and the other piece of DNA will have a T, T, C, and so on, and then it'll have a G. And these here will be loose, if you like. The bases will not be paired up. So you end up with what's called a sticky end. Why is it called a sticky end? Let's have a look on the next page. Alright, so a sticky end cut means that if these two pieces of DNA were ever put back together again, and an appropriate enzyme added, then the complementary base pairs would join up again. So A would join to T, A to T, T to A, T to A, and you would join the molecule back together. In other words, they stick together and can rejoin. So this is a way of actually joining together two bits of DNA that have been cut by a restriction enzyme or restriction endonuclease. This is very, very useful for joining DNA from different species together, and it has proved a very, very useful tool. Some of the other common restriction enzymes that are used, HAE3 cuts a blunt end, BAMH1 cuts a sticky end, so that can be useful, as we see here. Hindi3, as it's pronounced, also cuts at this sequence, and you'll notice that the particular base sequences in each case are different. And so it can be used to join different pieces of DNA from different organisms, depending on what the common joining sequences are that's available.